Now starting, all attendees are in listen-only mode.
Hello, everyone. Good morning. On behalf of Fight Colorectal Cancer, this is Andy Dwyer, and I want to welcome you to our October trial update webinar with featuring of the NCI-MATCH. We're very excited to have all of you joining us. I think we had almost 130, 140 folks register for today. So we're really excited about having such a great feedback in response to today's session. So we're going to go ahead and begin. I just want to say thank you for all of the great work. I know that with our founder, Nancy Roach, Dr. O'Dwyer, and Nancy have had inroads for a lot of work around the research advocacy and specifically thinking about access to clinical trials. So I think NCI is one of those that offers a lot of exciting opportunity. So we're really excited, Dr. O'Dwyer and Diane, to have you join us. Dr. O'Dwyer, would you like to introduce yourself?
Sure. So Diane and I are joining you today from ECOG Akron, and we really appreciate your dialing in for this presentation of the MATCH trial. It's, we view it as a key trial nationally, and your participation is key. I'm a medical oncologist. I'm at the University of Pennsylvania, and I am also the chair of the GI committee in ECOG Akron, and was previously the vice chair of ECOG Akron until recently. This MATCH trial was set up as a collaborative effort between the cooperative groups and the National Cancer Institute. They came to us to initiate this study, and as we go through the presentation, I'll tell you a little bit about it. And just let me thank you for this invitation to present today. Anne?
Good morning, everyone. I'm Diane Drago. I am the Director of Communication for the ECOG Akron Cancer Research Group and have been involved since the beginning of the NCI-MATCH trial, and I'm excited to have this opportunity to talk with you today about the latest news of the study, which opened in August of last year and is rapidly accruing, as Dr. O'Dwyer will explain as we move forward. So welcome.
Awesome, and it's fantastic to have you both. Just a quick reminder as we're getting ready to launch into today's presentation, we do just want to remind everyone that we do help support information and education for patients related to access to treatment, different types of opportunities for clinical trials, but of course no way or form any replacement for direct medical care. In order for all of you to be in contact with your medical team or 911 if there are specific emergencies. But we're really, really excited again for the presentation from the two of you today. I know we have a lot of people very much interested in the MATCH trial, as we are with all of the clinical trials work. So I think we're ready to go ahead and get started. I think Sharon, Michael, from the technical side, we look like we're good to go.
Yep, we're good to go. So we'll go ahead and move the slides forward. Thank you, everyone.
All right, Peter and Diane, take it away.
Okay, next slide. Thank you very much. And actually, I'm reminded that I just remembered that I didn't see a conflict of interest slide in here. So let me first of all mention about conflict of interest that I am funded for a large number of clinical trials, both through the University of Pennsylvania, ECOG Akron, and PRECOG, with a large number of institutions, and I consult for a number of companies, including Genentech, Amgen, let me see, 47. I think that pretty much covers it, and only Genentech, I think, has a compound in the MATCH trial. Of course, my difficulty in remembering that tells you how much of a conflict that involves. Next slide. And I'm here representing my co-chairs on this study. The principal investigators are Keith Flaherty and Alice Chen, one from ECOG Akron, the other from NCI, and myself and Barbara Connolly as co-leads. and Stan Hamilton, Mickey Williams, and Bob Gray, who are the former two have developed this exquisite technical tour de force, really, for sequencing of large numbers of patients and getting the results back in short order, and Bob Gray performing real-time statistical analysis of this study as it goes forward. A huge amount of work. Next slide. So what is this study? Of course, we all genuflect precision medicine, but this is really precision medicine in action. It was initiated with the idea that patients' tumors vary in a way that's determined by the genomics of the tumor, and that variability determines their response rate to various therapies and opens up the possibility of response from therapies directed to the genomic aberrations within the tumor. And so the idea was to accrue a large number of patients, large enough so that we would be able to test compounds in a significant number of molecular subgroups of cancer. Forget about where the cancer came from. The idea was to be agnostic about the cancer and just to focus on this genetic aberration, whether the treatment would work in that setting. The goal was to initially was to accrue 3,000 patients, subsequently expanded to 5,000, and in each patient to do a biopsy after the disease had progressed on standard therapy, to take that biopsy and very quickly extract from it the DNA in the tumor, analyze that DNA using next-generation sequencing, and if one found an aberration within that sequence that would be appropriate for a particular therapy, that the patients would then be offered the opportunity to have that therapy and to see if it would work for them. Next slide. Next slide, thanks. And so the sequencing is, as I've shown here, you take the DNA, ably represented with this cartoon, and extract it from the tumor and sequence it. Now, the sequencing is called next-generation sequencing, and it relies on a platform that was developed for this study by Stan Hamilton and Mickey Williams, and is robust. It looks for 143 genes. And in addition to that, a number of fusions and amplifications are detected. And as I said, if the tumor has a genetic abnormality that matches one of the drug subgroups, the patient can be eligible to join the treatment part. So let's distinguish two parts of this trial. There's step 0 and step 1. Step 0 is where the biopsy is obtained and the tissue sent to the relevant labs. Step 1 occurs if a match, a potential match, is identified from the sequence, and the institution practice is then notified that there is a potential match, and an opening on a particular treatment protocol is offered to the patient. Next slide. So the history of this is that the trial started with 10 arms in, when was it? April. April of 2016. No. Diane, help me with the date. When did it?
August 2015. Mm-hmm.
Right, and then the idea was that after the accrual of a certain number of patients, we were actually going to stop it after 500 patients, and take a look, see what we've got, see if we've designed the trial appropriately. As it happened, in a period of 3 months, we accrued 800 patients, and the trial was subscribed at a level that we would never have anticipated. It did better than any cooperative group trial in history in terms of accrual. We knew that in the initial part of the study that the match rate would probably not be very high because there were only 10 arms. And and that was true. It came out that the match rate, the potential match rate was 9% in that group. We had this pause in the trial, which was planned, and we took the opportunity then to make sure that additional arms were activated, and we activated the trial again with all of these arms on the 31st of May. And the trial has continued to accrue briskly since then. And with this expanded number of arms, the expected match rate is 1 in every 4 to 5 patients, and the actual match rate with our latest data that we discussed this morning is 23%. So there's close to a 1 in 4 shot of getting the treatment from this. Contrast this with other basket trials that have been done. It's actually an order of magnitude higher. And the idea, I think I mentioned this before, that patients with tumors that have the same genetic abnormality, irrespective of whether they might be breast cancer, they might be a sarcoma, they could be pancreatic cancer, they will receive the drug that targets that abnormality to test the question, is the key issue here just inhibiting what we conceive of as a driver mutation? If you can inhibit that driver that we think is driving tumor proliferation and the malignant process, does it even matter what organ the tumor arose in, or is that in fact an important consideration? Next slide. So the objectives are stated here, and the idea was to know if you could match a drug and a DNA aberration in the tumor, would that be effective? Would that be useful in treating the cancer, regardless of the cancer type? We emphasize that the NCI-MATCH is a signal-finding trial. It's not expected that a registration information would come out of this, but what we would like to find is signals that particular drugs and particular aberrations can usefully be put together and move forward. And obviously treatments that do show promise can advance to larger and more definitive studies. Next slide. Here are the eligibility criteria. I'm not going to go through them, but I just want to make a couple of points. This is for adults. There's a children's trial that's in development. This is for solid tumors or lymphomas, but actually we're pretty close to having myeloma ready to go. And I think these patients are currently eligible, or they will certainly be eligible in the near future. We had a focus on rare tumors. We're hoping that we've capped the accrual for the big four being colon cancer, prostate cancer, breast cancer, and lung cancer. We've capped accrual to each of the groups so that we can get a diversity of tumor types in there. And the performance status has to be reasonable. And one of the issues is that it does take time to get the next generation sequencing done. In my clinic, it takes 4 to 6 weeks if I send it off to a commercial assay. But the turnaround time for this trial is actually quite amazing. More than 50% of the results are back within 15 days. So we have implemented now encouragement. It's not in the protocol, but we're encouraging physicians to only select those patients who could tolerate being off treatment for the up to 6 weeks that it might take to get everything done. And that includes the time to get the biopsy, the time to process the biopsy, get the results back, and to do whatever tests are needed before a new treatment can start. Next slide, please. Sorry about this cartoon, a bit hard to read, but this is important because it kind of gives you the big picture here of the whole study. So, as we said, in the yellow box on the left, as we said, the patients get the biopsy. The sequencing is done. T10 is an immunohistochemical marker that actually allocates patients to one of the arms. And so if there's an actionable mutation, then the patient, and remember that's now 23% of the patients, can be matched potentially to a study agent. There are specific eligibility criteria for each of the study agents, as you can imagine. Some of them cause abnormalities, say, of cardiac function, and therefore there are cardiac-specific criteria for some of them. Some of them might put the liver at more risk, so there are liver criteria. And as you know, I mean, all of you know on this call that that's a feature of most of our therapeutic studies. So the patients who are eligible for the study drug are then offered the study drug and treated. And then in the gray boxes, you see the results of treatment. They get scans at intervals, usually 8 weeks or so, and the results of those scans will tell us how they're doing. They may have stable disease or a complete or partial response, in which case, obviously, we're winning. Let's keep on the treatment. If they have progressive disease, then there are a couple of options. One is to see if they have additional actionable mutations, because many tumors harbor more than one type of mutation. And we've set up a whole system for prioritizing those mutations in the trial. If they have an actionable mutation, then they could potentially directly enter that study if they're eligible. If there are no actionable mutations, then they're off the study and they look for other therapies. And then on the patients who are progressing, they obviously continue to until they're, the patients who are not progressing, they continue until they're progressing, and then there are a couple of options. Next slide. Here are the wait times. These are the current, most up-to-date times. So when a patient is registered, it takes 7 days to get the tumor sample from the site to MD Anderson. Now, that is way better than we expected. That is tremendous performance at institutions, at practices. This study is open in thousands of places, and it shows a very high quality of ability throughout the system. Then it takes the lab 13 days median and I'll show you all the data for that in a minute, to get the sequencing done and to return the results to the site. Part of this function involves a pathologist taking a look at the sample and making sure that it's the right disease. Just a detail. But obviously that takes time too. And then further eligibility evaluation occurs, and from the time of return of the data to the patient getting the treatment is a median of 14 days. And that's really why 6 weeks provides a pretty good time frame for time from registration to getting on the study. That encompasses the vast majority of patients. Next slide. . Here are the dates. Excellent, thanks. Can't remember this stuff. So we opened at 10. We paused 3 months later for the planned interim analysis. We continued to develop the treatments, and we expanded to 17 arms even before the trial had reopened, because we went back through the database and reevaluated patients who'd already had biopsies, and some of those patients were able to get on new arms. And then on May the 31st, we activated the trial for new patients with 24 treatment arms. Next slide. So this is the enrollment for treatment. We have 230 patients. Each treatment arm is seeking to enroll 35 patients. On a steering committee call this morning, it was clear that two of the buckets are filled at 35 patients. A number are also going in that direction, are very close, and then some of the molecular subgroups, somewhat surprisingly to us, actually have very few aberrations detected, very few patients who fit those criteria when expanded to the population at large. So we learn in different ways going forward with a trial like this, and this is part of the importance of the trial. Next slide. So the distribution of this, when you look at the distribution across the country, it is everywhere. It's in community hospitals and oncology practices, as well as at the larger cancer centers. And more than 50% are in the community. We're continuing to have new sites join. There's some restriction. All sites have to be willing to accept the CIRB. It's not available outside the US for similar regulatory reasons. And the list of institutions is on cancer.gov, clinicaltrials.gov. And Diane, I think it's on the ECOG Akron website also?
It is not.
It's not. Okay, so those are the...
Just the federal databases. Mm-hmm.
Right. Next slide, please. Oh, there you see it. Okay. Pretty good distribution, as you see. Next slide. Now, these are the 24 treatment arms, and the bars are what we predicted as the prevalence rates. These are not the numbers we started off with, because remember, we had this interim analysis, and we found that some of our assumptions going in were wrong. Some of the data from TCGA largely, TCGA is the big genomic effort to sequence cancers, and it's actually one of the best databases that exists out there for trying to assess how commonly a mutation is found. And we started off with TCGA and a number of other databases, and then the interim analysis came along, and we had to refine our suppositions, change our assumptions. But here are the assumptions that we have. And interestingly, the Tacelicib trial at the top for PIK3CA mutant cancers, you see that that's the most common, and that's also the one that crossed the line first. It's the one that completed accrual of 35 patients faster than any other trial. Right behind it, and also across the line, is Arm W. You see 3 down, the AZD 4547 trial, and that too has completed. And in between those, we expect the CCDN1 amplification to be a very common aberration in cancer, but because it started later, it has some catching up to do in terms of accrual. So you see that some of these percentages are, they're not high, and we are close to halfway through the study at this point in terms of total accrual. So that becomes a big issue in terms of the bottom half of this table. And we've explored a lot of ways, and this was obvious from the interim analysis. And one of the ways we've explored is that for the rare mutations, could we accept findings from some of the big sequencing companies such as Foundation Medicine? And we're almost ready to announce agreements with companies for that purpose. Next slide. They show the enrollments, and these again were based on the interim analysis and going to a total of 5,000 enrolled. And you see the numbers. The expected enrollment is shown in red. And again, these match pretty closely the bars that I showed you in the previous slide. And you see the importance of some of these subgroups, such as the especially E, F, V, A, and G. These are reported in various databases as being 1%, but we're not seeing 1% in this study. So some of them may not get to full accrual. Slide. So how do we find those rare mutations? And this is our strategy. A lot of our cancer centers, including the University of Pennsylvania, they're performing tumor testing with their own platforms, and we have engaged the cancer centers to use this as a funnel for accrual to match. We're also, as I said, collaborating with commercial sequencing labs, and of course, because this is an NIH trial, there will be a certain amount of red tape to be accomplished in defining those collaborations, but there's willingness on all sides to help, and we expect that these two strategies are going to increase the accrual. What I remind my colleagues is our goal is 35 patients in each subgroup, but if we were to get 25 patients in a particular subgroup, well, that's pretty good. That would be highly informative. Next slide. So our conclusions from the interim analysis, which guided our current iteration of the study, was, first of all, the trial is feasible. It can be done on a national scale, and it has really strong buy-in. We're currently accruing at between 115 and 135 patients per week. We expected about 50 patients per month. So this is remarkable how the cancer community has viewed this as a way of moving cancer treatment forward for patients. The whole process of tumor characterization is feasible, and actually that 87% number, it's in the industry, it's about 80% is pretty good. We're above 93% right now. And there's a high proportion of less common malignancies. And we think that's good because that addresses the principle upon which the study was built. Next slide, please. The interim analysis helped us to amend the study as we expected to do. And now we have planning for the realistic needs of additional trials and drugs. We're actually at a point where we're starting to limit the addition of additional subgroups because you know, the study is definitely going to close in 2017 in some way. And, you know, getting a study up and running is a challenge. It's not something that can be accomplished overnight. It requires the buy-in of a large number of people, review at CTAP, and activation through CIRB. So these are all considerations that make us kind of step back right now and have our largest focus is on two things. Getting replacements into the arms, replacement therapies into the arms that are accruing fast. And we expect to have that done with the next amendment. And the second is strategies to increase the accrual to the subgroups that are lagging. Next slide. So this is the summary of changes. I'm not going to read each one. You can see this. We've expanded the accrual. You know, it may be that the study will go beyond 5,000 patients. We're talking about that now. And but how it would be focused, you know, that there doesn't seem to be much sense in going beyond 5,000 without having a clear plan. And I think that it would probably need to be focused after that. We have lots more arms that are close to activation. Communication is important, and again, that's why I had highlighted in red that we would prefer that no additional therapy is given after registration on this study, and if the appropriate patients are selected, that's probably going to be okay. And we've added lab capacity, but actually we're doing just fine right now. Other changes here are to require needle aspiration because that increases the success of sequencing. To allow, in certain cases, tumor samples to be obtained up to 6 months prior to registration. There's a limit to this. It relates to what therapies they may have had in that 6-month period, and to allow data from other genetic platforms. Next slide. These are this again is at the point of 1,700. So our interim analysis was at 795 patients. So this is approximately twice this number. And you see that only 36.5% of the patients are in the common cancers category, whereas the less common cancers are represented by 63%. So that's the kind of spread that we think is going to increase the value of this study. Next slide. So I told you about the accrual rate. This is now 94% average, and the assay for bone marrow specimens is going to allow us to accrue myeloma patients. Next slide. I think that might be the last one, is it? No, here's the, okay, these are the resources, and these are important. We have on the main web page here at the ECOG Akron website, we have a frequently asked questions tab that covers most of the issues that we've addressed as part of this. And so if I don't answer your questions properly, or if you need additional information, please refer to that, and Diane will also be on the line to help us with that. Next slide. Next slide, please.
Peter, I believe that might be the last slide.
Oh, okay. So let me stop there. I tried to go through them a little bit more quickly to leave time for questions.
Great. Thank you so much, Dr. O'Dwyer. Sorry about the slides there. Are you still able to see that last slide, the resources for MATCH?
Yes, that's the slide.
Yes, we are.
Wonderful, great. So we did have a couple questions come in, and I'll go ahead and read those off to you. So we did have a question, and this is specifically relating to patients looking for clinical trials. If the question is, if I bring up MATCH with my doctor and they don't know about it, what should I do?
Oh, so I think you bring I think you bring two things. You bring the printout of the MATCH design and so on from the ECOG Akron website to an appointment. And I think also you see what resources are geographically close to you that might be able to help. One of the issues, before I advise patients to be traveling around the place and have potential hardship, one of the issues is to try and determine if one would be eligible for the MATCH trial before trying to find a way onto the MATCH trial. And I wonder, let me see if Diane has thoughts about that.
We have found that the National Cancer Institute's Cancer Information Service, which is 1-800-4-CANCER, is the easy to remember telephone number, has been very helpful to patients in this situation. They do have an accurate and up-to-date listing of the locations where the trial is approved and it's open and enrolling patients. And the advice and the regarding the study locations can be very helpful to patients in that regard. And I think calling the 1-800-4-CANCER information line, or you can even, send them a text message or an email, or even chat with them from the cancer.gov website, is much more of a means of assistance to patients than trying to navigate something like clinicaltrials.gov, where there's a listing of locations, but it's hard for a patient to go through. There are over 1,000 sites, 1,000 hospitals and cancer centers now participating in this trial. So it's a huge number of sites. So we do find that the Cancer Information Service is really the best resource for this type of information.
I also think that, I totally agree. And I also think that this is where the colon advocacy groups can really perform a service in that kind of linking way. And I would be happy to talk further offline about how such links could be made, how we can help you, and how you can help the patients for this type of issue.
Great, thank you. Next question. Does any arm of MATCH trial address KRAS non-wild type? KRAS?
KRAS non-wild type? Is that what you said? KRAS, yes. KRAS non-wild type? Oh, okay. So KRAS mutant patient.
Yes.
You can flash up the data. My memory is that if you flash up the bar chart, go back to whatever slide had the bar chart. I'll be able to take a look at that. Go back. Yeah, that's good. So yes, this is the issue. So if you look down about 7 here, binimetinib, there's a proportion of patients with colorectal cancer that are NRAS mutant, and those patients are eligible. For KRAS mutant patients, we actually don't have anything because the data are very preliminary on what might help. The incorporation of immunotherapy is obviously of interest in that patient population, but nothing is quite sufficiently credentialed at this moment to be included in the MATCH trial. We don't have an arm for that.
Okay. And next question, let's see. How many patients have been screened in total, and do patients and their doctors get all of the genetic test results back?
Total screening now is close to 2,000, might be a little bit over. The genetic screening data come back, yes, to the patients, first of all to the doctors, I think, yes, directly to the doctors. So all of the information that we obtain from this biopsy that might be useful for a patient to find a different trial comes back, and it comes back in real time because we recognize the importance of finding a trial if the molecular studies would indicate that to be appropriate.
Is there any specific efforts to be sure that there is ethnic and racial diversity and recruitment within this trial?
Yes, there are a number of efforts. I'll let Diane talk to that. And in part, this represents the map, the broad opening of this study across the country. But Diane,
yes, if you look across the 1,000 or more sites that are participating in the trial, many of those sites are involved with the National Cancer Institute's minority and underbased community programs, and we have, because of those sites being involved and having the trial open and available to patients, we know that the trial is accruing patients within those communities across the country where there is a predominance of either minority or underserved populations, such as in rural areas of the country. So there has been an effort made to ensure that those sites have opened the trial, and I don't recall exactly how many of the 1,000 sites are in minority and underserved areas, but it is a good representation, I think, of the trial in those areas.
Great, thank you. Another question, briefly, what is the difference between NCI-MATCH and the TAPER trial?
Oh, so I haven't looked at the slide set for the TAPER trial for a couple of months. There is some commonality of interest. I think that the key. Let me see if I remember the TAPER trial well enough. My sense is that the TAPER trial doesn't have a uniform platform upon which every patient is treated. So the eligibility criteria for the subarms of that study derive from data that are collected with a variety of platforms. The reason we thought it was important to have one platform for all the patients here is that we know that there's variability among these platforms, and I won't get into the technical parts of that. It's not the fault of the people doing it, it's just the fault of the technology. It's a characteristic, I should say, of the technology. So the TAPER trial also, though, is it's a worthwhile effort. It provides opportunities for patients to get onto studies that they mightn't otherwise be on their radar screen.
Mm-hmm. And this is Diane. I would just add to what Dr. O'Dwyer said. The TAPER study also does not have its own customized tumor testing platform, which is what we have in MATCH, as you heard. TAPER is considered more of a registry for patients than a straightforward clinical trial. because of the differences we described. So we know, for example, that in many of our hospitals and cancer centers around the country, they have both the NCI-MATCH trial open as well as the TAPER study. And in that way, if a patient does not have a match, and as you heard, 1 in every 4 to 5 patients will have a potential, will have a match to one of these mutations in our trial. But if they don't have a match, the treatments that those patients go on to will be determined by them and their physicians. The TAPER study gives those sites an opportunity to put the patients in the registry in TAPER and give them the therapies that they and their physician determine are the best approach, and monitor them and also contribute data to the TAPER study while the patient is on that trial. So it's a great opportunity to collect additional information about the patient and their treatment to put in a large database and provide to researchers, down the road so that they can study what's happening in these advanced cancers.
Great, thank you. I think we have time for maybe 1 or 2 more questions. Dr. O'Dwyer, can you remind us, is the trial open also for first-line therapy?
No. So this is a trial that's designed to kick in when standard therapies for the disease have been exhausted. So if there's something that really could confer a benefit to the patient in terms of survival, in terms of outcomes, we haven't thought it appropriate to have the MATCH trial to be, to be interfering with that particular therapeutic option. But we're talking about colon cancer here, and let me get to an issue that has been very important, and we've had dialogue about this internally, and have had dialogue with people who've sent us emails to ask us questions about this. So what the eligibility criteria for step 0 say is that the patient, I'm not looking at it right now, but what it says is that the patients should have had effective therapies for their disease. In colorectal cancer, what does that mean for a patient with metastatic disease? There are the standard therapies of oxaliplatin 5-fluorouracil with a targeted therapy, or 5-fluorouracil with irinotecan with a targeted therapy. And there have been new targeted therapies that are purported to have a small benefit of survival. Our feeling has been that the spirit of this eligibility criterion is best met if the patients have had an oxaliplatin-containing regimen and an irinotecan-containing regimen as part of their treatment. Or maybe they have a reason why they couldn't have one or other of those. And then they could be entered on the MATCH trial. So we're positioning this in colorectal cancer as a third-line therapy. I hope that will help in figuring out some of the generalities that are necessary in describing eligibility criteria for a large number of different kinds of cancer.
Wonderful. Thank you so much, Dr. Dwyer.
Okay. Sharon, are those the end of our questions for today?
Yep, we are just about at time. So thank you so much, everyone, for your questions, and Diane and Dr. O'Dwyer for the wonderful responses and the great presentation.
We really appreciate the opportunity.
Perfect, thank you. And I do notice there were just a couple other comments in the chat box that we can be able to definitely pass that information on to Dr. O'Dwyer and Diane about feedback about some of the trials and information and questions relative to the mutant KRAS. discussion and maybe some.
Yeah, we'll handle them. Shoot them over.
Okay, perfect. That sounds great. So we want to just, again, thank everyone and just remind everyone that if you do have specific questions about the clinical trials, specific information that you want to ask Dr. O'Dwyer or Diane, let us know. And if we'll probably go ahead and do some additional follow-up on the TAPER study, as well as some additional resources that are out there that we know that are very expansive, and go ahead and hopefully feature some of those trials just like we've done today with Dr. O'Dwyer and the MATCH trial. So just a quick thank you again for all of the time, the efforts, following along with us with Twitter, as well as following along on Facebook. Just a quick shout out that on November 8th, we are going to be featuring Heather Hampel, who is part of our medical advisory board, to talk about genetics, family history, and you, particularly getting a lot of questions from folks about the role of genetic testing, what to do with those results, inclusivity, basically findings of those sort of different types of therapies, and the findings from those genetic tests, and to also understand about what our next steps in terms of using those results, and if things are found to be inconclusive, where to go next. And so that's a lot of what we're hoping to feature with Heather on that presentation, and very excited to have her on board. And then, of course, any other additional questions, feel free to get in touch with us. Any other suggestions for talks, we're always open for the feedback. So Sharon, I think that closes up today's primary topics.
That does, yes. Thank you so much, everybody. Have a wonderful day.
Thank you.
Thank you.